For 4,000 years, the woolly mammoth has been defined by its absence. Since its extinction, the only physical evidence of the species has been the fragments preserved in the frozen earth. Colossal Biosciences plans to change that. By 2028, the company aims to have woolly mammoth hybrid calves living on the Arctic tundra. The technical framework was established by Harvard geneticist George Church, who first proposed sequencing the mammoth genome to engineer a hybrid mammophant. The scale of the project is massive. Colossal has raised over $400 million in total funding, making it the first decacorn startup in Texas history. The combination of significant investment and gene editing is changing the timeline of extinction. These resources are being used to turn a permanent loss into an active restoration project. Creating a perfect mammoth clone is currently impossible. After thousands of years in the permafrost, ancient DNA is heavily degraded, shredded into tiny fragments that cannot be fully reconstructed. Instead, scientists use a living scaffold. The Asian elephant shares 99.6% of its DNA with the woolly mammoth, providing an exact evolutionary template. Using CRISPR technology, geneticists splice specific cold-resistant mammoth genes directly into the Asian elephant's genome. They target sequences that code for specific survival traits: a 10-centimeter layer of insulating fat, smaller ears to minimize heat loss, and thick, shaggy hair. This circular diagram tracks the de-extinction process via precise hybridization. It tracks the pipeline from digital sequencing to physical cellular research. The project requires taking the code analyzed in silico on a computer and physically inserting it into living cells in vitro. This process results in a hybrid proxy, an Asian elephant specifically engineered with the traits required to survive in the freezing Arctic. Splicing the genes is only the beginning. The next hurdle is taking those engineered skin cells and turning them into a viable embryo. Researchers reprogram elephant skin cells into induced pluripotent stem cells. These blank slate cells multiply rapidly and are directed to carry the edited mammoth DNA. To grow a calf, these embryos could be implanted into living African or Asian elephants. However, using endangered species as surrogates carries immense biological and logistical risks. To avoid using live hosts, Colossal is experimenting with artificial wombs. These are massive, fluid-filled tanks lined with engineered uterine tissue. Building a physical environment capable of growing a 200-pound calf is a massive challenge. Overcoming this gestational bottleneck is just as critical to the project as the genetic editing itself. If these engineered animals are successfully grown, they serve a specific purpose in the wild. The Arctic permafrost contains enormous amounts of trapped carbon. As global temperatures rise, this frozen soil melts, threatening to release greenhouse gases into the atmosphere. In winter, large grazers trample snow while foraging, removing insulating air pockets so sub-zero air keeps frost from reaching the soil. Herds also knock down trees. Dark forests absorb solar radiation, but vast grasslands increase Earth's albedo, reflecting sunlight back into space. The mammoth proxy is being designed as a biological tool. By restoring these grazing patterns, the herds could help repair the ecosystem and prevent a massive release of greenhouse gases. This demographic chart illustrates the divide between standard conservation and de-extinction. Traditional conservation maintains an unbroken lineage of living animals. De-extinction requires forging an entirely new genetic lineage to replace what was lost. This creates friction in the scientific community. Many conservationists question spending hundreds of millions on an engineered proxy, while living elephants struggle to survive poaching and habitat loss. The Ecology Law Quarterly highlights another concern. Wild populations of a de-extinct species would likely be geographically restricted and highly vulnerable during their early years. This leads us to a difficult question. By releasing these engineered hybrids into a modern habitat, are we restoring a lost ecosystem or introducing a well-funded invasive species? Human activity is currently driving a global mass extinction event. At the same time, the capacity to rewrite the genome is a reality. The tools required to edit and synthesize DNA are rapidly scaling. This leaves us with a central dilemma. We can focus our resources on the fragile ecosystems that remain, or use these genomic capacities to try and replace the biological engines we previously destroyed.